the T tubules. And then this kind of pale stuff that looks like somebody spread gum around is the sarcoplasmic reticulum. You need to know the organelles of contraction. Those are the myofibrils, and that the myofibrils consist of a series of sarcomeres. The sarcomeres extend from Z disc to Z disc with the thin filaments of actin attached to the Z disc and the thick filaments of myosin in between. That's everything you need to know on that model. Okay. Next models I'm going to have one or more of these guys. So this is where you have to distinguish amongst the three different types of muscle. This one is cardiac muscle. It's got striations, it's got intercalated discs, it's got branched cells that could have one or two nuclei in them. And of course, it's under involuntary control. And or I might have this guy. This guy is smooth muscle. It's got the tapered ends. It doesn't have striations. It has a single nucleus, and it's under involuntary control. And then finally, skeletal muscle. These are the long, they're said to be straw-shaped cells. They have striations depicted in red, but no intercalated discs. And they have multiple nuclei depicted in black here. And these are the ones that are under voluntary control. So everything you need to know about those cells. Okay. It looks like I've got about 12 different individual skeletal muscles for you to identify. I promise you I'll have both legs, both arms, the head and neck muscle, and then the muscle man. For the muscle man model, I'm going to use the one that's got the blue tape up there. Okay, the old one, it's just falling apart and the technician keeps removing it. I'm pretty sure he's going to throw it in the rubbish before I know it. And so when you look at the key, it says old and new, and I'm talking about this one when I'm talking about the new one. This is the new one. I haven't gotten around to this guy yet, figuring out the numbers. So this is the guy you want to look at. They look similar enough, though, that both should be able to help you with your studying. Okay, so 2 legs with 2 questions on each leg, 2 arms, 2 questions on each arm, and a couple questions on the muscle man, a couple questions on the head and neck, and then don't forget the eye. There were the rectus muscles and the oblique muscles of the eye. You will have that in there. That would be your easy one, right? Okay. Questions about joints. There's some questions about movement and then joint classification questions. So I could do something like, go into the bone box and pull out a skull and point at a suture. And then I could ask, what's the structural categorization or what's the functional categorization? I have a whole bunch of synovial joints up there. Those are really easy. They're all structurally synovial joints and they're all functionally diarthroses. So expect a handful of those as well. And I would refer you to the chart that I presented in lecture and also to the coloring. So any examples of specific joints would come from those 2 items. Okay. All right. Okay. For the endocrine system, I've got multiple models for that too. Just to review, this one over here. The only thing I'm going to use on this model will be this part of it with the hypothalamus and posterior and anterior pituitary, or this part with the parathyroid glands. All these other guys I have elsewhere. So I've got the male and female reproductive anatomy for the gonads, great. I've got the cut open adrenal gland for the adrenal cortex and medulla. I've got the thyroid gland on the torso over there. I've got the pancreas on the torso, and then I have just a roaming around free living pancreas with a spleen and a little bit of duodenum attached to it. So there's only 2 things you need to pay attention to here. Okay. Also with the endocrine system, we studied a couple of slides. We looked at the thyroid gland with the follicular and parafollicular cells, and we looked at the pancreas with the islet cells. So for the pancreas, you just need to know about the islets. We're not going to have any questions concerning the acini cells. Okay. Then for reproduction, we also looked at slides. We looked at young male testis. So we focused there on Leydig cells, on spermatogonia, mature spermatozoa. Those are the only things you need to have a handle on for that slide. For the mature follicle, the Graafian follicle, we just really focused on that secondary oocyte with the zona pellucida and the corona radiata around it. Don't really have to know too much else about it. For all of the endocrine system stuff, just like I said in lab, you need to know the gland, you need to know what it secretes, you need to know the target of that secretion, and you need to know what that secretion does, what is the action of the secretion. Okay. There are going to be 4 questions that are fill-in. I'm so nice to you. The 4 questions that are fill-in are from the male and female reproductive anatomy models. Okay. The terms you need to know are in the lab manual. Even though it's a really crummy list, it's not very complete, I will stick with that. So there's terms in bold under short activities for the endocrine system lab. I will take terminology from that or terminology of things that you have colored. So make sure you know all the terms that you colored in the coloring book. Okay, so you have 4 questions. If you can use the speller, I've got the spellers back there and I'll have them out like before. So it might behoove you to look in the speller tonight and find out where those things are. Okay, then 10% extra credit will be to draw out the steps of the sliding filament model of muscle contraction. So while you guys are starting to work on your muscles, I will stand up here and jot that down real quick. So I'm going to do it as bullet points. So instead of doing it as an essay, it's easier for you and easier on me and better to grade if you just do it as bullet points, step 1, step 2, step 3. Now last night we had a discussion about this, that do you want to say that ATP is hydrolyzed and then there's a signal from a neuron, or do you want to say that there's a signal from a neuron and then ATP is hydrolyzed? I'm okay with it either way. I like to point out that the ATP has to be hydrolyzed so that the tissue is ready to contract, so I put that first. But if you switch those 2, that's okay with me. But then everything else should follow a sequence. Acetylcholine is released, the T-tubules depolarize. This causes the sarcoplasmic reticulum to release calcium. Calcium binds to troponin. That makes the tropomyosin shift. That opens up the binding sites for myosin and actin, and then myosin can cross-bridge with actin and shorten the sarcomere as it releases its stored energy. Easy peasy. Easy for me to say. So I'll put that up here, and so you can take a picture of it and you can try to memorize it, but if you understand it, it'll be a lot easier to spit out on a piece of paper. So then when you come next Wednesday, you'll need a Scantron, and then you'll need a spare piece of paper to write out that sliding filament model. Everyone good with that? Rachel's good.